Hello and welcome to this video on differential gene expression analysis. We will see what the results from the bioinformatic steps of RNA-seq are and how to call differentially expressed genes. My name is Marc Galland and I am a support data analyst at the Swammerdam Institute for Life Sciences at the University of Amsterdam. This lesson we explain you several things. First, you will be able to describe the two inputs required for a differential analysis using DESeq2 package from R. First, we will see the gene counts matrix that summarizes how much each gene is expressed in each sample. It consists of a lot of rows, one per gene, and a lot of columns, one per sample. Secondly, we will see the sample to conditions table that links each sample to its experimental condition. For example, samples that belong to the same condition as the control condition will be related and shown in this table. You will then learn how to describe the table of differential genes obtained from DESeq2, in particular, what each column refers to, how to filter results to only get the meaningful differentially expressed genes. You will also see how to interpret a classical figure from an RNA-seq differential analysis. This figure is called a volcano plot, and we will see in details in this video how you can describe it. You will also learn how to select top candidate genes and how to use additional information from external expression databases to yield a biological interpretations of a gene different. There are also more advanced topics, but they will not be in this video, but in a later one. So first, let's see a workflow overview. How do you what do you do after completion of the bioinformatic steps of RNA-seq experiments? So here you can see a complete overview of the sequencing and bioinformatic steps of an RNA-seq workflow. It goes from sample collection, library preparation, to the sequencing of the isolated RNA. This will give you a series of FASTQ files. You will then deal with the bioinformatic steps of an RNA-seq. So first, you will quality check the reads. You will remove the adapters from the sequencing reads, assess their quality as well. Then you will align these reads to a genome using splice-aware aligners such as STAR. And finally, you will count the reads associated with the genes with software such as feature counts. So from the sequences present in a FASTQ file, these sequencing reads, depicted here, which are present in the FASTQ files, will be aligned to a genome. So this is represented here. And the aligned reads aligned to the genome will be counted. So in this section, for example, the blue reads will be counted as belonging to gene A, which consists of 2 exons. And this gene A will get 6 counts of 6 because it has 1, 2, 3, 4, 5, 6 reads aligned to its portion. Same for gene B, which where you see the green reads aligned to the exon of gene B. It has 4 reads, therefore the gene B will get a total of 4 counts. So the counting procedure will generate a count matrix, and this is the one that you will use for differential expression with DESeq2. So all these steps from making the library, sequencing the reads, until counting the reads for the genes will be the bioinformatic section of the RNA-seq pipeline, while the count matrix and the data analysis will happen in R, and these are downstream steps. So all the bioinformatic steps usually take place on the computing cluster using the command line interface and some ad hoc open source softwares. On the other hand, the differential analysis in R usually takes place on a local machine, a desktop or a laptop. So let's see the first input that we'll need for our differential expression analysis, the gene count matrix. So this is the first input. It's usually referred as the gene count matrix or the row count matrix, has several different names. So what you see is the first column is usually the gene column, and it contains the identifier of the genes whose abundances have been estimated. The following columns are the samples and are listed here as sample O1, sample O2, etc. In each cell, you will find values. So in the first cell, you will have, for example, a count of 2. That means that this gene was estimated with 2 reads in this sample. It was detected 2 times in sample O1. Some genes will have only 0 counts, so not like this one, but imagine the one here and one here are zeros, then this gene will not be detected, but still you will have a complete row of zeros for that gene if it's undetected. And some genes will have a lot more counts than others. That's also something to remember. Yeah, that's what I was telling you. The second input for the differential expression analysis of genes with DESeq2 is a sample to condition table. So the sample identifier are present in the first column, so so not in the column names, but as rows. So sample O1, sample O2, sample O3, just like the same names as in the input number 1. And on the second column, you will see experimental factors. So here you have only one called genotype. And this experimental factor has 2 levels, either wild type or knockout, for meaning that it's a mutant. It has a mutation somewhere. So if you would have other factors, you would add them as additional columns. And usually you have 1, 2, sometimes 3 experimental factors. So you will have sample, genotype, another condition, condition 1, and another condition, condition 2, maximum. So now that we have the 2 necessary inputs to call differentially expressed genes, let's have a look at what the general principle is and how to perform it. So what is described here is the DESeq2 methodology, but other softwares are possible, but they will slightly differ. If you consider a single gene expression coming from different samples in 2 conditions, it can be depicted as such. So on this scheme, each dot is a sample, and it shows the expression level of a gene, so from low to higher. So the more you go to the right, the more expresses your gene. The blue dots on the left hand are representing the expression of the gene in the condition A, and the red dots are depicting the expression of the gene in condition B. And what you see is that the blue dots and the red dots are quite separated. So if you consider the global mean of all these genes, the group means of condition A, and the group mean of condition B, you can see that the mean of condition A and the mean in condition B are quite separated. So they deviate from the global mean. And that would mean that for this gene, there's a significant difference in the gene expression due to the condition. So the gene expression in condition A and condition B are not the same. On the other hand, if you take another example, we see that the blue dots and the red dots overlap a lot, and that would mean that there's no significant difference in the gene expression in the 2 conditions. Well, what really happens in DESeq2 is more complex, but if you keep this simple example in mind, you'll get an idea of how it works. The main difference is that we're dealing with thousands of genes at once and not just one gene. So in R and using the DESeq2 package, all this estimation of differential expression happens in mostly 3 steps. First, you import your data, so the input number 1, the counts, input number 2, samples to condition table, and you provide a design, so a formula. How do we want to model the counts according to your experimental design? So here we had one condition called genotype, so this is going to be our design. You then feed this object and you allocate it to the same object using a function from DESeq2 called DESeq that performs the differential analysis. It does a lot of things under the hood. And finally, you will be able to extract the table of results using another function called results. This result table looks like this. So you will obtain a table with a few columns. What is first important to realize is that all the genes are kept in this result table, not only the genes that will be considered differentials later on. So first you have the base mean, which is here. So the base mean is the mean of the normalized gene counts in all samples, regardless of their condition. So it corresponds to the global mean that we've seen before. A high base mean infers that the gene is globally highly expressed across all your samples. Then we have a log2 fold change here. So that's the log2 fold change is the log2 transformed fold change between condition A and condition B. Then we have this LFCSE, which stands for log fold change standard error, and that's the error associated with the estimate of a fold change. So the higher this value is, it means that the fold change has a relatively higher uncertainty associated to it, but it's harder to estimate it correctly. Then we move on, we have stat, which is the test value, a wall test, which is similar to a t-test for large values of a number of samples. The uncorrected p-value, so the raw p-value corresponding to the wall test value, and you need to correct it because you're performing multiple tests at once. So this is why you have the adjusted p-value at the end of the table. So that's your first output, but it's a table, so it's not quite graphical. So what you can do is make a so-called volcano plot, which would be the output number 2. And there are multiple ways you can do that. And one way is to use an online application listed here in this reference. So this is the exact representation as the table, but in a graphical way. So the x-axis here will represent the log2 fold change. So center at 0, and here it goes up to minus 2.5 up to plus 2.5. On the y-axis, you have the adjusted p-value significance. It's plotted on an inverted log 10 scale so that you can visualize it better. This representation is useful to visualize the number of up and down accumulated transcript in response to an experimental condition, a treatment, a mutation, etc. So you can see if a volcano plot has a balanced shape, so if you have similar number of down-regulated genes or up-regulated genes. It is also useful to highlight interesting genes. So in this case, you can see that some genes, some points have received gene names and colored. So according to some criteria, so they have to pass a log2 fold change threshold here, and also a p-value threshold, which is also here. Okay, but what to do next? How do you go from a list of differential genes to some biological interpretations and insights? So first, you have to remember that from the DESeq2 differential analysis, you get a table with all the genes, not only the one that you can consider differential, but also the ones that are not differential. So you can easily verify this by counting how many rows/genes are present in your table. If it's in the range of 20 to 30,000 rows genes, that means you have all the genes in your table. So therefore, you will have to filter this table to keep only the genes that you consider differentially expressed. This relies on choosing 2 thresholds. So first, you have to choose the absolute log2 fold change that you want to select. So this is from this column. This relates to how strongly regulated is your gene. So the threshold has to be an absolute fold change because negative values will correspond to down-regulated genes and positive values will correspond to up-regulated genes. The second threshold to choose is the adjusted p-value threshold, so this column. The lower this p-value, the less likely you are to find false positives among your differential genes. Remember, you have to select on the adjusted p-value because you're performing multiple tests at once, and that means they need to be corrected. If you have a low, if most of your log2fold changes are small, let's say minus 0.5 plus 0.5, there is no point of choosing a threshold of minus 2 plus 2, because you might lose most of your affected genes. So the threshold has to be chosen based on your data set. That being said, most publications will choose a log2fold change threshold of minus 1 plus 1 or minus 2 plus 2. That's usually the case. As a side note, a log2fold change of minus 1 corresponds to a fold change of 0.5, and a log2fold change of 1 corresponds to a fold change of 2. So this means they are equidistant, so the log2fold change of minus 1 is a reduction of half, and a log2fold change of 1 is a multiplication by 2. When you have filtered your genes, you can isolate the most promising genes. You have filtered the result table to select only the differential genes based on the two thresholds, log2fold change and adjusted p-value. And for now, your table of differential genes all have an absolute fold change higher than the selected one, and a p-value lower than the selected threshold. But still, you will probably dispose of several to thousands of differential genes. And therefore, you will have to select the most promising ones in order to investigate them using online databases. So the three main criteria you can use for that are three criteria. So the first is the absolute log2fold change. So the higher the better. The higher this number, the stronger your gene expression is affected by the experimental condition. The second criteria is the adjusted p-value. So the smaller, the more significant is the chance that your observed changes are related to the experimental design. So this is a sort of confidence index. Another criteria is the base mean, because the mean gene expression of a gene across all conditions will be linked to that number. So the higher that number, the more expressed is your gene. And once you know that half of the genes usually have a very low expression, this can be useful to filter them. It's a bit subjective, but what I would recommend is to filter in this order. So you would rank first on the absolute log2fold change, followed by the base mean, and eventually by the adjusted p-value. Once you have your list of the most promising genes, you can gather information online. So some databases are generic, such as the David database shown here. So this allows you to gather annotation on a gene list, convert between different identifiers, and query multiple other databases at once. So here I have an example of a gene from Arabidopsis with its short name and gene symbol, and all the links that redirect to other resources. So indicated here is the KEGG pathway, which is a database on metabolism, and you have a link for fatty acid elongation. So you will see the gene in the context of a metabolic pathway, and you can actually directly go on the KEGG website because there's a link. In the case of Arabidopsis thaliana, for instance, you can use specific plant databases like this one called ePlant. This is actually a web service that gathers information from other plant-specific databases, and it renders it on the same website, in the same portal. And this will give you a comprehensive view of the gene expression in multiple tissues, genotypes, etc. You can refer to the Introduction to RNA-seq video to see a more comprehensive description of its databases in action.